Hi everyone, it's great to be here. So my name is Lendert, and I want to start by showing you a few snapshots of a developing mouse embryo. Okay, so this is a developing mouse embryo, and I think development is really one of the most incredible and interesting things to happen in nature. You go from a single cell and you end up with this full animal. And if you look at these snapshots, and you look at the complexity of tissues, organs, a brain to think that this animal has, I find it kind of hard not to think that there's some very sophisticated guidebook that is guiding this process, or that is the blueprint of this process. But it all starts much simpler, at least with a seemingly simple process, but it's very crucial. The moment that you go from a single cell to two cells. And so here I'm showing that for the same mouse embryo. And what the cell is doing, here in red you can see the DNA, and the DNA is getting copied into two copies, and then it needs to be split between two daughter cells. And it's done using these blue fibers, which are called microtubules. They take the DNA and they pull it apart, and then the cell splits in two. And so then you've gone from one cell to two cells, and if you do that several times, quite quickly, you have quite a lot of cells to work with. If we were to do that about 30 times or so, we would end up with billions of cells, and that's about the number of cells we've got in our body. Now, unfortunately, my clicker is not working. Now, there we go. So unfortunately, it's kind of hard for a mouse embryo or for a human embryo to visualize that process live because it's internally that it's going on. But we can do that for various other animals. I'm thinking about fish, frogs, salamanders, and here I want to show you a movie about the early development of a salamander. And so here you can see the egg, and it's incredibly big, these salamander eggs. They're 2 to 3 millimeters in diameter. So you can just see it with your bare eye. And the nice thing is that you can see the process just by eye. Another thing that is quite nice about this process is that it is very fast. So you don't have to be too patient to see the development occurring. So I'm going to show you here these first divisions. And so what this embryo does, or the strategy it has, is really just divide, divide, divide as fast as possible. So you go from 1 cell to 2 to 4 to 8 and so on. And it does this as quickly as possible, and so you end up, after 10 divisions or so, with a few thousands of cells. And this is a very fast process and the fast part of development of this early salamander. It really wants to just go as fast as possible and cleave one big cell without growing the cells as fast as possible in many smaller cells. Okay, then we go to a next stage of development. And that's the movie that I'm going to show you here. Oh, there we go. So what happens then is that the process of development slows down. The cells, they don't try to make, or the embryo doesn't try to make as many cells as possible anymore, but rather it starts positioning and moving the cells where it wants to be. So you got this ball of cells, and it folds in on itself, and the embryo starts making various decisions. What is going to be my inside? What is going to be the outside of my body? Where is going to be my head? Where is going to be my tail? Right? And so this process then develops, and all the different cells also start making decisions in which type of cell they're going to be. For instance, some cells become muscle cells, others become skin cells, yet other cells become nerve cells. And as you start watching this movie, you can see the shape of the embryo evolving. You can see a head, you can see a tail, you can see organs developing, and at some point, this embryo will get out of its shell, and it will hatch, and then you've got a larva, and this larva is then free to explore the world and to interact with the world and swim away. All right? And so that's what's going to happen any moment now. But if I look at this movie, I wonder, how does every cell know what to do in this embryo? how does every cell know where to go? Or in other words, how does life work? And this is the next slide that I've already shown you, because this was a recent book, which is called How Life Works by Philip Ball, and I can recommend it. It's a great book. It also influenced a bit of my presentation. And, well, yeah, so that is one of the questions we will look at today. But it also connects to an even deeper question, which is, what is life, to start with? And this question was really made famous by Erwin Schrödinger, who is a scientist, he was a physicist, and he's mainly known for quantum theory. So not for biology, but yet he wrote this small book about what is life. Now, he was not the first one to do that. This question has been very influential for many years, and it's still a question that is very much alive today, and even Nobel Prize winners, such as, for instance, Paul Nurse, who works at the Crick Institute, and he got the Nobel Prize for figuring out what the key molecules are that control cell division. He also had a go at this question. Now, I'm not going to answer this question today, because way too deep. I'm not equipped to do that. And I want to just focus on some key aspects of the What is Life book by Schrödinger, and two main ideas. But before I do so, I want to have a quick detour, just because maybe Schrödinger, his name doesn't ring a bell immediately, but it might do if I show you this, because he's also known for a famous thought experiment where there was a cat in a box, Schrödinger's cat, and this cat is in a box, and it's together with something that can kill it at any moment, but we don't know when. So from quantum theory point of view, this cat has to be described as being both dead and alive at the same moment, which is a bit weird. So he had this thought experiment to show the weirdness of the quantum world. But that's all quantum mechanics that I will talk about today, because I want to focus on Schrödinger's work when he was thinking about life. And here you've got a picture of chromosomes, because when he got interested in the field of biology, a lot of things were already known, and people had been looking under the microscope at cells that are dividing, and in fact, they were looking at salamander cells, the cells that I've been showing you before. And what people noticed is that there are these dark thread-like structures that are copied inside the cell and are then pulled apart and before they're split into two daughter cells. And those are chromosomes. And there was a biologist, Walter Fleming, who described this process in great detail, and he called this process mitosis. And it comes from the Greek word mitos, which means thread, so hence mitosis, the process of cell division where you split the two thread-like structures between the two daughter cells. So back to what is life. So Schrödinger was influenced by these observations, and what he wanted to know is if you have these chromosomes and they're split between two cells, it's a sort of inheritance that is given to the two daughter cells. So what is the information that is inherited and how is it encoded? So he made a suggestion, and the suggestion was that the information is stored in a so-called aperiodic crystal. It's a solid structure that can contain a complex code, and it's not just a repetition of a periodic signal, all right? And basically, we know now what this aperiodic crystal is, and you all know it. It's DNA. So it was discovered, the structure of this aperiodic crystal, by Watson and Crick, and also by Rosalind Franklin, and a contribution by Morris Wilkins. And there's another second idea in What is Life book, and that is that life needs energy to stay organized. So basically, Schrödinger was thinking originally about quantum mechanics, so the way that the physics works at the very small scale. So he was used to thinking about particles moving, fluctuations, and he wondered how can it be that at a small scale everything is fluctuating, is jiggling around all these molecules, and yet at a large scale, like ourselves, we are very much ordered beings. And we're all familiar with this problem because normally life goes typically towards disorder. If we don't clean our kitchen every day, or our bedroom, or whatever, well, it will quickly explode and will be a mess. So we actively have to put energy in it to keep it organized. And that's the same thing in biology. You need to put energy in it to maintain the certain order. And so this is also something that Schrödinger described, and it led to a type of non-equilibrium physics, which was also pioneered here in Brussels by another Nobel Prize winner, Ilya Prigozhin, who really described this. So in short, there are two main ideas in Schrödinger's book, What is Life? That is, well, you've got this aperiodic crystal, so life needs information, and it needs to be organized, and it needs energy to do that. And so it was mainly his first message about the aperiodic crystal that was very influential in the scientific world. And it also came to be that Watson and Crick also saw his book, and in the 1950s they discovered the double helix, the structure of DNA, and they wrote a letter to Schrödinger saying that they were very much influenced by his little book, What is Life, and they wanted to share that they discovered the structure of DNA, which according to them was this aperiodic crystal. So I would like to serve you 3 cocktails today. Okay, and each cocktail will take about 10 to 15 minutes to consume together. And if it's too much information, or if you're drunk on information at some point, you can call me out and say, well, we should take a break, and I'll be happy to do so. So we're going to explore 3 parts of life. We're going to look at a DNA cocktail, a cytoplasmic cocktail, and a cocktail of cells. And it's not just a metaphor here that I'm using just because here today we're at Science and Cocktails. I'm actually literally meaning that we're going to make some cocktails today, and we're going to see how we can take DNA outside of the context of a cell, similar for the cytoplasm and the cells, and put them all together in different ways to see how they behave and how they organize themselves. So we're basically going to do some mixology here as well. So the first cocktail is the DNA cocktail, and we're going to do that by using strawberries, or I'm not going to do that right now, although I tried at home earlier today. But I'm just going to use this video. So strawberries have a lot of DNA in them, and if you squash them, you can break it up. And what you do is you add some pineapple juice, which contains enzymes that break down certain proteins, so the DNA can sort of loosen up. A next step in preparing this DNA cocktail is adding some strong alcohol. The reason to do that is if you have this strong alcohol, the DNA is not going to stay dissolved anymore, and it's going to come out like these pale, stringy threads, which I think you can see around here appearing. And so the reason I'm showing you this is because I want to show you that this DNA, it's a physical thing. It's a physical thread, literally, that is really big. In a typical human cell, if you would spread out the DNA, you would get 2 meters of the stuff. So it's a physical thread that you can now see with your own eyes. That ball there is DNA. And so you can see it, and if you want, you could also taste it. Okay. So this DNA, it's a double helix that Watson and Crick discovered. It consists of 2 big strands that are combined and intertwined, and it's a bit like a zipper where you've got 2 strands of DNA. And it consists of a quite simple alphabet. It's got the letters A, T, G, and C that are repeated one after another. And the reason that it's a zipper and you can open it up and close it up and they can find each other is because these letters combine with each other in a certain way. A will connect to T, G will connect to C. So if you open it up, you can copy the DNA because they can match each other and you can get a new strand of DNA. Okay? And so it was around 2000 that they really were aiming to sequence the whole genome of the humans, and you had the Human Genome Project, and they reported on that in the year 2000, that they had read the whole genetic code of humans. And I think it was then President Bill Clinton who gave a speech about this, and he said, well, we now have the whole human code, and he compared it to the findings of Galileo, that who also said, like, well, we have now the language in which God created the universe, and now soon with this human code, we will be able to understand life and cure disease. So big words, and people were, and rightfully so, very enthusiastic and excited because it was a big achievement, of course. And so here's an example of this piece of code that contains A, T, C's, and G's. And we can look at it, and now 25 years later, we can ask ourselves, is this now the key to life? Is DNA the blueprint of life? And to some extent, we've learned things. For instance, there are some very peculiar or well-defined diseases that are just related to a single change in the genome or a single gene. So a gene is a part of the DNA, and there's just a single mutation. An example is sickle cell disease. It's a blood disorder related to hemoglobin, and you could target that disease by, for instance, gene editing or gene-based therapies, and in that way you can solve it because it's a very well-defined part of the DNA that you need to fix and is directly related to a known disease. But in practice, things typically don't work that way, and diseases are much more complex. If you think about cancer, for instance, it starts with mutations in the genome, either due to aging or exposure to UV light or what have you. So it starts with DNA, clearly important, but then the disease itself, it's much more based on how the cells interact with each other, how they form a collective tumor, and how they respond to the immune system. So the most important advances there that have been made, or some of the advances, are more related to immunotherapy, for instance, where you use the cell, the human's own defense system to attack the tumor, right? So here's the classic picture. We've got DNA that contains our information, that is transcribed into RNA, which is then translated into protein, and those proteins create a certain behavior in the cell or have a certain function. And this is called, by Watson and Crick, the central dogma in biology. And it's true, I mean, this does happen, but I think there's a problem calling it a dogma because it's a bit misleading, because a dogma means that you cannot question it. This is the whole truth and nothing but the truth, and we should not question it. So I think it's the fact that it's called a dogma, and also that a lot of books were written that were focused on DNA. Think, for instance, about The Selfish Gene by Richard Dawkins and other books that really place DNA very central and portray it as the blueprint of life. And I think there are a few problems with that. There we go. So in order to figure out some of these problems, I want to do a little quiz, and I don't want you to think too long about it, okay? So which creature here has the largest genome? So if we look at the book of our genetic code, who has the biggest book? Who has the most letters? Is it a human who thinks about how life works? Is it a frog who thinks about what he's going to get fed, hopefully soon? Is it an amoeba, which is a single-celled organism that, well, I don't think it thinks about anything because it doesn't have a brain. But okay, so we've got these three organisms, and I want to see a raise of hand or vote. So who thinks the human has the biggest genome? Okay. Who thinks the frog has the biggest genome? Oh, well, okay. Who thinks the amoeba has the biggest genome? It's quite evenly split, actually. Pretty interesting. So, well, I don't know. There are a lot of votes for the amoeba as well, for the single-celled organism, which is funny. It might also be because I was maybe a bit suggestive with the question, or I was leading the witness a bit. It could also be that I just chose a very bad sample to represent humanity. There were also several votes for the frog, which perhaps is because, well, the talk is a bit about frogs. I've shown you these movies of salamanders, which have huge eggs, so maybe a lot of DNA and a particular type of development. And then there's, of course, the human. So the first surprise here is that both the frog and the human have 3 billion base pairs of DNA. So there's not really a difference, and that is already strange. It's not tracking with what we may think or what our intuition may tell us about complexity and how DNA relates to complexity of an organism. And then if we look at the amoeba, it has a much larger genome than us. It has 200-300 times larger genome than us. So basically, we have to see that, well, genome size rather does not really correlate with complexity of an organism. So let's get back to this picture of the central dogma. And as I said, it's true, right? You go from DNA to RNA to protein to function. But there are a few issues with that. And the issue is, or one of the biggest issue I think, is that to go from DNA to any of the other stuff, you need life already. So to go for DNA to a living system, you need a living system to achieve this. It all takes place inside a living cell already. If you want to, the DNA in the end is just a text, it's a genetic code. So you need the machinery and the molecular machines to read that text, to process the text, to correct the text, and to maintain it, right? And so that's why you need a living cell. Apart from that, once you're inside the cell, there are other things that go on. For instance, you've got proteins that can bind to the DNA and regulate their own expression. So you've got not just this nicely directional linear view, but it's a whole web of interactions. It's much more complicated. And then finally, this all occurs inside an environment. If you have the same DNA inside a similar cell, then it will behave differently depending on the signals it's receiving, the nutrients it has available, the temperature it's experiencing, and the environment it's in. So I want to change this picture and go more to the fact that, well, DNA certainly matters, but it exists inside a living cell, inside a complex network, and inside an environment. And this is very much in line with a book called The Music of Life by Dennis Noble from 20 years ago, where the main message was that there's no single privileged level of control in the cell. So the DNA doesn't have a privileged place. And he had a metaphor or analogy where DNA is not the music, but it's the music score, and you need a musician or an orchestra to get anything out of that music score. So same thing with the DNA, it only makes sense in the context of a living system to get something alive out. Okay, and that already brings me to my second cocktail, which is a cocktail of cytoplasm. We started with the DNA, which is information, and here the cytoplasm is basically all the stuff and the machines that you need to make sense of that information, right? And so we're going to start by doing something a bit strange here. We're going to make a cytoplasmic cocktail, but we're going to remove the DNA. I don't know what it's going to do, right? Is it still going to do anything? Is it still going to organize itself? I don't know. This is what we're going to try to figure out. And the star of the show here is our African clawed frog, the frog Xenopus levis, which you've seen in the quiz before already. And, well, it had quite a lot of votes, actually, so I have the feeling it's reasonably popular. Now, we're not going to work with the frog itself, but we're going to be working with the frog eggs. And just like the salamander, it has very big eggs. It's about 1 millimeter in size each egg, and it lays a lot of eggs, hundreds of them, every time the mother lays various eggs. Okay, and this is what an egg looks like. Some might think it looks like the moon, but if you fertilize it, it comes alive, and it starts dividing with incredible precision and regularity. All the cells divide every 20, 30 minutes or so. And so to see how this works, we can look inside of the cell, or inside of all of these cells, and look at the cytoplasm, what it looks like. And it typically would look something like this. It's an incredibly dense place with a lot of molecules that are all densely packed. A different picture we could look at is the following, where you've got tons of proteins or pieces of RNA or other things, and you can connect them with all kinds of arrows, meaning that they're all interacting with each other and influencing each other. So it's a very complicated network of interactions that controls the behavior of the cell. Now, the nice thing about using and studying these early frog divisions is that it's a bit simpler. They go very fast and they're very regular. They're essentially controlled by a nice biochemical oscillator or a molecular clock. And that clock can be described not just by, it is influenced by the whole network, but to capture the essence, we don't need all of it. And we can just zoom in on a key part of it. There we go. A small part is enough to understand how this molecular clock works. So we forget about this whole spaghetti of interactions and we just focus on this very simple system. And it starts with RNA. Inside the egg, you've got already RNA that the mother provided inside the egg. It's stocked with it. And that RNA is used to make protein, many proteins, but I'm thinking now about one particular protein. I'm not going to give you the name unless you ask me later, but a protein that controls cell division. There's one key enzyme that really leads to cell division, okay? So that is made continuously, and as soon as you have a lot of that protein or that enzyme, then it triggers cell division. Now, at that point, it also does something else. It triggers cell division, but at the same time, it also triggers and activates the machinery that degrades itself. So this is called a negative feedback loop, and this is how this clock works. Basically, synthesizing, you make the protein activates, leads to cell division, and then it leads to its own degradation, it's reset, and this is the whole clock. Right? And it's so simple that we can make mathematical models of it. We can write down equations for how these proteins are made and how they're degraded, and we can put that we can put parameters to it from experiments, and we can simulate it in a computer. And it looks something like this, and it's something what you would hope to see. Basically, it's a signal that goes up and down in a rhythmic fashion. And every time the signal is high, the cell will divide. You can compare it with another biological oscillator, one that you are likely more familiar with. Right? If it's an oscillator, just our heartbeat. It has a similar periodic signal that is contracting our heart. And the person who described the first model for these cardiac rhythms is Dennis Noble in the 1960s. And this is the person who wrote the Music of Life book, which I showed you before. And the nice thing about this is that the principles that control our heartbeat are almost exactly the same as what controls the cell division. You also have this simple negative feedback loop where sort of voltages rise and fall, and you get reset, and you've got your cardiac rhythm. The difference is that the key players are different. We don't have RNA and protein that is made. Rather, we have ions that move across a membrane and go up and down. So let's really go into how we make our cytoplasmic cocktail right now. And it starts with a frog egg, which is stocked with everything that you need for early development. It's got DNA, it's got energy, it's got a lot of cellular machinery, such as ribosome, mitochondria, endoplasmatic reticulums, Golgi apparatus. You name it, it's in there. You don't need to know the names, it doesn't matter so much, but it's stocked with all of that stuff. And so what we do is, basically, we don't make a cocktail like the strawberry pineapple daiquiri we had earlier to look at the DNA. It's more like a pisco sour, where we break open the eggs and we make the cocktail like that. And the way we break open the eggs, we don't use a blender like we use to make a smoothie, but we use an ultracentrifuge, which is this big device. And inside this ultracentrifuge, you put a rotor, and inside this rotor, we put a tube with all of our frog eggs. And this device does not use any blades to mix it. It just spins really fast. And because it spins so fast, it generates an incredible gravitational force. So the very heavy elements, they get spun outwards, and the lighter elements, they're not spun so much outwards. So basically, you're separating all of the content of your frog eggs by weight. Okay? And because it's a cocktail, I guess you can also describe this as something that is shaken, not stirred. So here you can see the device in action. So it spins really fast, and you get these 3 layers out. You get a top layer, which are lipids, is the lightest layer. Lipids is what makes up the membrane of each cell. Then the heavy part is the yolk. It contains all the nutrients for the embryo to survive. And then the middle layer is what we will use, is the cytoplasm. So we take this middle layer out, and we use all of the components inside this. So it could be all of these machinery that I've discussed before. Now we have this cytoplasmic extract, or this mix. And what we do is we try to make it as realistic as possible to mimic a real cell. So we make these bubbles of extract, and many of them. And so they're sort of artificial cells, and then we look at them under the microscope. And if we look at them under the microscope, what we see is the following. We see that they're flickering, all of these. So we completely mixed it, but yet they're flickering every 30 minutes or so. And the 30 minutes rings a bell because it's exactly the same period with which all of these cells divide. So basically what we're seeing in these artificial cells that we make is an oscillator that says divide, divide, divide. So we've taken this biochemical clock outside of the context of the cell, and we've recreated it in these artificial cells that we make. And here you can see a signal of that. You can measure many things in these cells, and the way we do that is we use fluorescence under the microscope, and you can color many different parts with many different colors. And here we use a color for this critical enzyme that leads to cell division, the one for which we made this model that I showed you earlier, right? And so here's the signal, and this is experimentally, and if I show you the simulation, it looks pretty similar. So I think that's quite nice. You get the same rhythm out, and you also even get the same shape of the oscillations of this biochemical activity of this critical enzyme out. And there's more surprises, because you don't only have a biochemical clock that you can make, but that biochemical clock can at the same time, in this artificial cell, organize spatial cellular structures. So here I'm going to be showing you a different color. Now the color will be so-called an actin biopolymer. Now, it doesn't matter what that is. What I want you to know is that this actin is critical in each cell because it's what splits each cell in two. It creates a force to split the cell in two. So if we now color this part of our artificial cell, and we look under the microscope, we see this contracting ring. And this ring contracts periodically. So basically, you also have this clock that is driving the periodic contraction of this ring that basically is getting the signal divide now, and it's trying to do so. It's creating a contracting ring which is trying to constrict the cell. And it's not working because we don't have a membrane that we built. Something else that we're trying to do, but that's not working yet. Next step. Another thing that we do is we add the DNA back in. And other cool things happen, or at least I think it's cool. So we put the DNA in there, and it's not just floating around in this extract, but what is happening is the following. So around the DNA, this extract is going to build an entire nucleus around the DNA by itself. And that's shown in green. So you've got a nucleus, and this nucleus appears and disappears periodically with the same rhythm of our biochemical clock. And this means that every 30 minutes, the nucleus is broken down, and the DNA it's copied, and they're gonna try to pull it. It stopped. Okay, I can start it again. So the red is microtubules, also another polymer. But what they do, those are the blue fibers that I showed you in the second slide of my movie. Those are the ones that pull the DNA apart. Okay. And so you've got the nucleus disappears, and then you've got this red that tries to pull the DNA apart. So what I think is amazing about these artificial cells that we make is that you've got a biochemical clock, but not just that. Spatially, it organizes all of the different cellular structures that are in a real cell. You've got a nucleus, you've got DNA that gets copied, you've got this actin that tries to pinch off the cell. So it's quite realistic. Next question I want to address. Imagine you've got this huge frog egg, 1 millimeter in diameter. The cells that I've been showing you right now, these droplets, they're more in the order of magnitude as our cells in our body. For instance, 20, 30, 50 to 100 microns. 100 is already big. So they're quite small. Now, these frog eggs are huge, so that brings also a problem of coordination. How is this huge egg going to divide whenever it receives the divide now signal? Imagine that this part of the cell, this enzyme gets activated, and it says, you have to divide now. How will the other side of the egg know that it needs to divide? Because it's way too big. On the size scale of these molecules, it's huge. So that is really sort of a coordination problem. And it's one that we've thought about, and people in biology in general have thought about, and there are solutions to solve this. I'm going to get back to the heart again. So the solution here in the heart is that you have a pacemaker in your heart. You've got a region in the heart that is beating faster than the rest. And because of that, it sends out a wave of contractions. And so all the cells, whenever this electrical wave passes, will contract in a coordinated fashion. And that's how the heart works the way it should work. An analogy you could use is these ropes, for instance. If you flick a rope, you've got a pacemaker, it's going to give you a wave that travels throughout the space. Okay? So this is an analogy you could think about. Now the question is this happening in the same way in this frog egg? And it's something we can test, because we can take our extract, our artificial system, and we can now, instead of in a small droplet, put it in this long tube of millimeters long. And I'm going to show you a movie of the result. And the color that you'll see is, again, this critical enzyme that drives cell division. Okay? And so what we see is that it activates at certain locations, and then it's spread like a wave throughout the whole tube, over and over again. So it tells us that, indeed, these waves help to propagate the information over these very large distances. And this is not just an artificial system or an artifact of the fact that we're using these extracts. Actually, you can see the same waves inside a real embryo just before it divides. So we're trying to uncover some of the physics that helps coordinate these early cell divisions. So that was almost a one-dimensional system, a long tube. We can do the same in a big droplet. And what you see then is these concentric waves that move outwards. Basically, like you throw a rock in a pond and they ripple outwards, right? So that's the typical signal that you would expect. But under some circumstances, the wave can break up and you can get these spirals. And I like to show this because I just think they're very pretty. But it also tells us something more. The spirals, they're actually also physiologically relevant. Again, the heart, because I think it's a nice analogy. If you look at the heart, you can also get these waves of electrical activity, and that's not good. That's basically associated to heart arrhythmia that you can have. You're disrupting the typical coordinated pattern of a beating heart, and when you have these spirals, they self-sustain and they mess around with the proper coordination of the beating and the pumping of your heart. What you can then do is defibrillation. It's basically a shock and a reset of the system so that hopefully you go back to your normal rhythm. Okay? So these spirals are physiologically relevant. And it's also logical that we see them because from a physics point of view, once you have these traveling waves in a larger system, you can expect to see them. And also in the frog egg, we believe that they could be physiologically relevant to disrupt the proper coordination of cell division. Okay, maybe let's breathe a bit. Short break. Everybody's still with me? Are there people that were here also last month for the How to Buy Happiness? Okay, good. I hope you bought your ticket a lot in advance so you can enjoy it now, enjoy the experience. I also hope that some of you bought maybe that space flight. Anybody? Because I would like to try that experiment with the extract actually in the absence of gravity. So if you did do that, please let me know. But okay. Let's go to the cocktail of living cells. Oops. So what I want to do now is look at cells and whole embryos, put them together in different ways and see how they behave. I want to use a similar cooking analogy again. For instance, you can think about caviar, and that's the picture that I had here. Whoops. Imagine you've got caviar and you've got all these beads and you can shape it into any shape you want, right? Now, depending, you can make this, but you can also make a pancake or a ring or whatever. But all of these different situations lead to a situation where the beads are connected in different ways to their neighbors, right? And so what we're interested in is if we change the shape of this structure early in development, how will that evolve? Will it develop into a normal tadpole or will it become something else? And of course, here in the analogy with caviar, these beads are actually live dividing cells. Right? And we can do that. If you have the typical frog embryo, you can take the membrane off. They're sort of inside of so-called vitelline membrane, and that keeps them in a nice spherical shape. And if you remove that, as is done here, they're free to take any shape they want, basically. And so these are type of experiments that we're doing. I'm going to show you on the left a normal embryo, and on the right an embryo where we remove that membrane. And so here you can see, if you remove the membrane, these cells are fine, they still divide, but you see that it looks very different. They sort of are this pancake shape now. They divide inside in a sheet, but yet, with time, they have a certain coordinated organizing response. And depending on how you do it, even if you put them in different shapes, they will end up in a tadpole and in a proper frog. But you could also do something else, and like here, for instance, it will develop into a strange shape, and eventually it will die. But the point I'm trying to make is that the shape of an embryo, we often see it as an end result, right? But it can also be a starting point or a constraint. The shape actually changes the way things will develop. For instance, if you have tissues, once they have a certain form, that form will also dictate which forces they will and which signals they will receive, and they will guide development in a certain direction. Now, can we model that in a way that is informative? And here, we're working together with the lab of Bart Smeets at KU Leuven as well, and we're creating these foam models. We're basically modeling the dividing cells here in the embryo as bubbles in a foam. And we're trying to do two things at the same time in these type of simulations. On the one hand, we have the mechanics. We have all the cells that are in contact with each other, like these bubbles in a foam, and you've got the mechanics of it and the physical constraint of how they can divide. And on the other hand, what we're putting in internally are these biochemical clocks and the models that describe these dynamics. And so what we're hoping to find is that we can create these type of models to predict how development will continue if we change internal properties or if we change the constraints of the shape at which they start their development. And so first simulations look like this, where you've got a normal embryo, and on the bottom you remove that spherical membrane, and they divide in the sheet just like you've seen in the experiment. So this is very preliminary, so I don't have a lot of interesting things to share yet, but these are just different directions we're trying to go into. Now, one step further, instead of just changing the shape of the embryo, another thing that we can do is we can really sculpt a whole body plan and cut out living cells and put them back in a different way. And so this is work by the lab of Michael Levin in the US. And they're the same, maybe I can play it again. They're the same embryos that we use, but you cut them open, you remove the membrane, and you take these white cells from the middle out, and you put them now in a different location together. Just like before, the cells, they're alive, and they will try to clump together, and they form this ball of cells. If you wait, this is what you will see. You see, they're coming together. And then once you have a ball of cells, you can try to make a different shape. So what you will see is that they cut it, and they make different types of shape out of it, different shapes out of it. I'm just gonna give it here, see? Now you're just gonna cut that, make the shapes that you want. It's a bit crazy, right? And so what's striking here is that if you get the shape right, and you can check that in simulations, you can make these things do things you would like them to do. For instance, move. And here's an experiment that they did, like, oh, here they designed it so that it moves, and if they turn it upside down, it doesn't move, showing that they understand how it works. Now I have to wait a bit, I don't. So, what makes sense now is if you can make these different shapes, is that you want to explore the whole space of possibilities of the types of shapes you can make. You can't try all of the shapes experimentally because it's a lot of work to make it by hand. It takes hours to do one. But in the computer, you can do these simulations, try one shape, and see whether it's good at certain tasks. For example, are they very good at moving? Or collectively, are they good at picking up garbage collectively? Other thing that they check is, are they good at taking cargo and pushing it around? And something that I haven't said yet, but I forgot, is that these things, they're basically like living robots that you're making, and they're called xenobots, because they're living robots made from xenopus cells, frog cells. And here was another example where the shape is such that it can carry cargo to different locations. Another nice aspect of it is that they're living things, and because they all organize themselves, they're very robust. So you can cut them and you can break them up and they will repair themselves. So we often talk as if life follows a blueprint, or that DNA has some kind of plan that an architect made that we then use to make life. But I hope that this evening I've tried to convince you that this picture is not really complete. Instead, they're often just interactions of many components. All of these molecules, they interact with each other. The cells, they interact with each other, following local rules, using energy and taking information from the environment. And they just organize themselves. So there's no blueprint of life. Instead, there's just the process where the living matter is organizing itself one step after another. And I want to end with this bigger picture, because we can use these extracts or embryos that we shape in different ways, and we can explore this space of possibilities. We break it down into components to a certain extent so that it still makes something that is lifelike. Right? It reorganizes itself to do things that we recognize as more or less alive. But we can really explore this whole space of possibilities. We can add multiple components. We can add more or less, just like a recipe that we're cooking with these ingredients. We can change the shape or the geometry. We can change the temperature and see what's out there. And so I think that is something that I really like, that we can explore that. And it teaches us something about, not just about how life is as we know it, but also about how life is as it could be. And I think I just want to end by thanking everybody in the group, the people who did all the experiments, the people who are developing the tools and doing the technology, the people who are making the models and the theory, and all of them are, what I appreciate is that they're combining biology, physics, engineering. And I also want to thank all of you for listening, and yeah, okay, go ahead.